Excellent. Thank you very much. So I'll try to keep the microphone up near the mouth. Okay, excellent. So, well, thank you very much for the invitation and the opportunity to speak here today. So I thought I'd tell you about some of the work we've been doing on lung cancer evolution and tumor heterogeneity, and also the interface between the cancer cell and the immune microenvironment. So although this might be somewhat difficult to see, and this sort of recapitulates a lot of what we've seen already, what we're interested in my lab is very much this intra-tumor heterogeneity, so diversity within tumors. So how can we decipher the extent of diversity within tumors? How prognostic is that, and what can that tell us about the evolutionary history of a tumor, and also the interface between the tumor and the immune microenvironment? So most of what I'm going to tell you today is using data from the TRACERx consortium. This is tracking non-small cell lung cancer evolution through therapy. That's what TRACERx stands for. And this is a multi-center study where we're looking from primary diagnosis through to surgical resection, metastatic disease. We're aiming to sample the tumor throughout its disease course. We now also have a linked warm autopsy study to this. So we're really aiming to explore tumor evolution in detail. Okay, there we go. And ultimately, we aim to sequence over 6,000 exomes from 842 patients. So we aim to have a lot of data here for quite a large number of non-small cell lung cancer, early stage patients, really to be able to get to the heart of can we identify a lethal subclone, and can we identify patterns in how these tumors are evolving? And we're doing longitudinal sampling, also doing cfDNA, CTCs, and having quite valuable clinical data associated with this as well. So initially I thought I'd tell you some of the data from the first 100 patients that we've looked at that we published just last year. So this is the clinical details for the first 100 patients. So we've got, here we can see that we've got 60 or so lung adenocarcinomas, and then we had 32 lung squamous cell carcinomas, and then a mixture of others. These are, for instance, mixed histology patients. And you can also look at the colors that indicate whether or not these patients were smokers or not, and the gray and the dark gray are ex-smokers and current smokers, and then the blue are the never smokers. And perhaps not surprisingly, most of these patients are quite heavy smokers, because this is lung cancer. And then we have a mix of males and females, really. And this represents a sort of all-inclusive cohort across the centers where we're studying this. And so how do we actually explore the heterogeneity? Well, as other people have alluded to, what we do is we use multi-region sequencing. So here, rather than take a single sample, we take multiple regions of each patient's primary tumor, and then we use deep sequencing of exome sequencing of each region, as well as the matched germline, to try to infer the mutations and copy number events within each region, and ultimately, as we've seen already, to try to infer a sort of clone tree and the clonal architecture of each tumor to work out which alterations are early events and clonal, and which alterations are subclonal, occurring later during the tumor's evolutionary history. And this is the data for the first 100 patients, where we've simply classified each mutation as clonal, so we think present in 100% of the cancer cells that were sequenced, and that's in blue, and then the subclonal alterations present in a subset of the cancer cells. And what you can see here, so each column here is one of the patient's tumors. You can see there's quite a large amount of heterogeneity in the extent of heterogeneity itself. So you have certain patients' tumors, like the one over here on the far left, where we have thousands of subclonal alterations found only a subset of the cancer cells, whereas we have other patients that have thousands of clonal alterations. And clearly this also depends on how many tumor regions we've sequenced, but in each case we've sequenced between 2 and 10 tumor regions. And you can also see there's some differences here, whereas we've got a greater range in mutation burden in lung adenocarcinoma, it's more uniform in lung squamous cell carcinoma. I think this largely corresponds to the fact that lung squamous cell carcinomas, they're pretty much all heavy smokers, so you've all the patients almost have quite a high mutation burden. Whereas in lung adenocarcinoma, we have some never smokers and also some heavy smokers, and we also have other mutational processes occurring in these tumors. So this is at the point mutation level, and then we can also look at the copy number level, and we've already seen that this can be quite extensive. So what you'd expect normally is 2 copies of every chromosome across the genome, and what we actually see in cancers is these are highly messed up genomes, and so this is just a plot here of one of the regions we've looked at in one patient's tumor, and we can see that actually in this case, a large proportion of the genome is present at 4 copies, so we think there's been a genome doubling event at some point during the tumor's evolutionary history. And this is one region, then we can look at the other regions, and we can start to decipher heterogeneity here too, to see events that are specific to certain regions and absent in others. So we can now add this to the plot. We've got mutations, then we can also look at copy number events, and we can see again there's quite a variation in the extent of heterogeneity at both mutational level and copy number level. So the first question we were really interested in with the TRACERx study is this prognostic or not? And sort of to put in sort of graphical form, we reason that perhaps if the tumor's phylogenetic tree looks something like a palm tree with lots of clonal alterations found in every cancer cell, then that patient might do better than if their evolutionary history looks something like a baobab tree with extensive diversity. So we thought, okay, well, this is really what we want to test. Let's do this in the simplest way possible. So first, looking at mutations, if we just split on that plot to heterogeneous versus relatively homogeneous according to the median proportion, do we see a difference? And so when we did this at the mutational level, we don't see any significant difference in survival at all. And I think this is partly as well that we're using a very crude measure of heterogeneity here, that we're also going back in time. So a lot of these mutations might have happened prior to actually carcinogenesis. So these patients at this stage, some of them will have received, well, afterwards will receive adjuvant chemotherapy and some of them won't. But when we include that, that doesn't really change things very much. So at the moment, they haven't received any targeted therapy. It's uniform platinum chemotherapy. Yes, so there is a relationship there, and generally the larger tumors will see somewhat more heterogeneity in, but it's at the mutational level, it's not quite such a simple measure. So it depends whether you're looking at a sort of clonal architecture heterogeneity or you're looking at proportion of mutations that are heterogeneous as well. So at the mutation level, I think we can go a lot deeper, and this is just our sort of simple, because this is what we specified before, we're going to just do the simplest measure possible. When we do that, we don't see any relationship. And when you start to go into a bit more detail, you do start to see relationships. But what we thought was also quite interesting is when we did the same thing but looking at copy number, we saw quite a clear distinction here. Yes, so we can also try to normalize where we take, for instance, the average of the two because we have a minimum of 2 regions, so we can also average there. But that doesn't make a huge difference for the survival analysis, at least. So in terms of copy number heterogeneity, we see quite a clear distinction. So tumors with more heterogeneous copy number events seem to do a lot worse than those where the copy number events, if they occur, they're homogeneous. And what I think was interesting as well is when we take a sort of static picture of copy number, so if we just look at the proportion of the genome that's aberrant, we didn't see any difference in survival here, suggesting it's potentially this ongoing chromosomal instability that's particularly prognostic in these tumors. And when this was significant in multivariate analysis when you include size and subtype and stage. So that really suggests that copy number heterogeneity is important for prognosis. And we want to look in this further. We're doing work also with Roland. I think we'll present some of this work later on. But next we can also ask if we've seen, when do the driver events occur? Are these occurring early in every cancer cell, or do these occur later on during tumor evolution? So this plot, I appreciate, is probably very hard to make anything out of at all. But if you're using your imagination as well, what it's attempting to show is here we're trying to work out the potential drivers in each of these patients' tumors. And in blue, we've got the clonal drivers, and in red are the subclonal drivers. Now, in every patient's tumor, we identify clonal drivers, which makes sense that there's initiating drivers. But interestingly, in 75% of the tumors, we also identify subclonal driver events. Then we can start to look at, do we see differences in what alterations occur early compared to those that occur late? And this is a very busy heat map showing the frequency events and then all the genes that we're considering as potential driver genes. And you can see there's certain events that occur at very high frequencies. This is your p53, your EGFR, your KRAS mutations. So here we can see that these tend to almost always occur clonally, but even these sort of very well-established driver alterations sometimes occur subclonally in a subset of the cancer cells. And we can again start to see if there's any patterns in general here in these cancer genes. And one of the things we see is when we look at the clonal driver alterations. So here I'm showing you the frequency of clonal driver alterations in lung adenocarcinomas. We can see that here we've got p53 and KRAS, and in red are the ones that we think are more specific to lung adenocarcinomas that identified in analysis of lung cancers. And we think these are the ones that are occurring at high frequency in the clonal events, which makes sense. But when we look at the subclonal events, we see these are sampled for a far wider selection of cancer genes. So we think potentially early on there's certain constraints to tumor evolution, and these change as the tumor develops, which I think fits with what we saw earlier as well, that there's a clear distinction between the early driver events and the later driver events. And I think we really don't fully understand the subclonal later drivers in as much detail as we understand the early driver events. And we see this pattern in lung adeno, and we also see a very similar pattern in lung squamous cell carcinoma as well. So this allows us to start to build up a model of how we think these non-small cell lung cancer tumors evolve. So here I'm just showing, it's a depiction of how we think this occurs. And we think in these tumors there's certain driver events that are frequently very early, and some of these almost always occur early, such as p53 in lung adeno and lung squamous cell carcinoma. Then we have KRAS mutations also occurring early in lung adenocarcinoma, but when they occur in lung squamous cell carcinoma, they tend to be later events. And we can also look at the mutational processes here. I'm not going to go into detail about these, but here we think early on we see lots of smoking-induced mutations, which makes sense because these are smokers. But then when we look at the later events and we can time these relative to genome doubling, we see a shift in mutational processes. So that while the early events are dominated by smoking, the later events we see other mutational processes often occurring. For instance, ApoBec-mediated mutagenesis takes over as a dominant process in many of these tumors. But one of the interesting things we can also see is when we look in the ex-smokers and long-term ex-smokers that stopped smoking sometimes 15, 20 years ago, we can still pick up smoking-induced mutations after a genome doubling event, which suggests that the genome doubling event might have happened 15 or so years ago, because we can time it relative. If we're still picking up smoking mutations occurring later, that suggests the genome doubling must have occurred while the patient was still smoking. So we think these tumors have been developing for a long period of time, particularly in the lung adenocarcinomas, whereas we think it's more rapid tumor evolution in lung squamous cell carcinomas. And then we can look at the later events, and here we barely see a smoking signature at all, because this is, as I said, dominated by other mutational processes. And here we see more alterations involved in DNA damage and repair rather than the classical drivers. So far I've focused very much on purely the genetics and the genomic events that we see in these cancers. The other thing we can also ask was the interaction between these events, because all these events occur in a given microenvironment. So can we start to look at which mutations might actually give rise to mutations that the immune system could recognize? So one thing we can do is we can actually start to predict whether mutations might be recognized by the patient's own immune system. So to do this, we can use a quite simple technique, which is not terribly accurate, but gives us some information at least. So for each mutation, we can predict whether it gives rise to peptide sequences that might be presented by the patient's own HLA molecules, and therefore might be recognized by the patient's own T cells. So for each mutation, we can predict its binding to the HLA molecule. And there's various tools that we can use to predict this, and this allows us to say whether a mutation might potentially be a neoantigen, so we can predict whether or not it might be recognized. And we did this initially a few years ago looking at TCGA tumors, particularly in lung adenocarcinoma. We tried to predict whether the mutations might give rise to neoantigens. And not only did we do this, we also tried to predict whether these neoantigens were clonal, found in every cancer cell, or subclonal, found only in a subset of the cancer cells. And we predicted that perhaps if a patient has more neoantigens, then it's more likely to be recognized by the immune system. and so it will engage a more active immune system, so therefore the patient's prognosis might be better. And when we looked at this, we saw exactly that. So patients in the upper quartile of total neoantigens, this is in lung adenocarcinoma again, seem to do better than those in the lower quartiles, or the remaining quartiles. But then we also asked, if we add a level of heterogeneity to this, do we see a clearer distinction in survival? And we do actually see exactly that. When we introduce an ITH threshold and specifically look at clonal neoantigens rather than just all neoantigens, we see a clearer separation of the survival curve, suggesting it matters not just how many neoantigens, but whether these are presented by every cancer cell or only a subset. So next we can also ask, what about in the context of immune checkpoint blockade? So where these patients have been stimulated with immunotherapy to try to unleash these T cells. Does the heterogeneity make a difference? Or in this case, is that not actually important? So there's data already indicating that the number of neoantigens is prognostic here in relation to immune checkpoint blockade. So we wanted to ask, well, does heterogeneity come into play here as well? So we took the data from that study by Rizvi, that they'd elegantly shown number of neoantigens as prognostic, and then we separated these neoantigens into the patients into the durable clinical benefit and no durable benefit. And then we could also identify here, just as they had done before, which is quite reassuring, that we see a difference in number of neoantigens, whether or not these patients benefit from immunotherapy. But then we could also predict whether the mutations were clonal or subclonal, even though we just had the single samples here. And what was quite interesting is we saw some of these clear outliers that have lots of neoantigens but do badly. Actually, a lot of these mutations are subclonal, found only a subset of the cancer cells. So this really seems to highlight that we really should be looking beyond just number, but actually at their clonal status as well. And when we look at the heterogeneity between those patients with a durable and non-durable benefit, we see a clear difference. And indeed, we see a very clear difference in survival between these cohorts as well. And we could also look at this in melanoma as well. So we could look at just number of neoantigens, and when we add an ITH threshold, we see a clearer separation. Well, I can just about see it. You probably can't quite see it, but you should be able to see a clear separation of the survival curves. And this is one cohort. And we also looked at one cohort that was published recently, where we had, again, responders and non-responders, and we worked out the clonal heterogeneity. And we saw some patients that had really a very high number of neoantigens, but no response, and a high number of subclonal neoantigens. So we want to ask, okay, well, what's actually generating these subclonal mutations? And why are we not getting any response here at all? So this is showing the plots where we've tried to break up the clonal architecture, and we're looking at mutations across the genome, and we saw lots and lots of mutations at really very low variant allele frequencies that initially we thought, okay, well, maybe we're just picking up sequencing artifact. So maybe actually they don't really have a high mutation burden at all. It's just that it looks like they do because there's lots of sequencing artifact. But what we could do is we could actually look at the mutational signatures, so look at the context in which the mutations occur to see whether this can indicate what's caused these mutations. So when we looked at the clonal mutations, these are melanoma patients, we saw a very clear signature which has been associated with UV. So this makes sense. These are caused by UV light, and these are the early mutations. These are melanoma patients. That makes sense. So what about the later mutations? Well, we looked at the later mutations. These also had a very specific signature, but in this case, we could actually decide that this signature looks exactly like a signature induced by alkylating agents. So in this case, it was actually these patients had received temozolomide treatment prior to immunotherapy, so it was the treatment itself that caused a lot of these subclonal mutations, which is a little bit worrying, where you're seeing all these mutations and these patients aren't benefiting from immunotherapy, but they're caused by their prior treatment. Okay, so the final part of my talk, I'd like to focus on one particular aspect of the neoantigen presentation and more about copy number heterogeneity in these tumors. So as I said before, one of the ways we can predict whether mutations might give rise to neoantigens is whether they bind to the HLA molecules. And we can predict the specific patient-specific HLA alleles from the sequencing data. And the HLA alleles it's a highly polymorphic region, and each HLA allele has very specific binding properties. So you have your maternal A, B, and C alleles, and your paternal ones as well. And each HLA allele, as I said, will have specific binding capabilities. So different neoantigens will be presented by different HLAs. So potentially one mechanism of immune evasion is to lose one of these HLA alleles through loss of heterozygosity. So this is simply where we have a loss event, so we no longer encode both the maternal and paternal, but we've lost one of these. And so this means that we no longer have SNPs in this region. So if we lose one of these HLA alleles, this means we might lose the capacity to present neoantigens. This might be one mechanism of immune escape in tumors. And indeed, this has actually been published in the literature that, in this case, there was a study looking at targeting the KRAS G12 neoantigen, which is found to bind to the HLA-C0802 allele. And upon relapse, it was found that this HLA-C allele was downregulated, so it seemed to be lost. So we said, okay, well, maybe this occurs more frequently even in the treatment-naive setting. So we wanted to ask the question, can we identify this HLA-LOH event? Is it a high-frequency event in tumors? And how frequently does it occur? And when does it occur during tumor evolution? And finally, if it does occur, what is the functional impact of this? So we first asked, well, can we easily do this? Well, normally what you do is you'd look at the coverage of heterozygous positions and look at your SNPs and identify regions of LOH. Well, as I said before, the HLA allele is highly polymorphic, which means our reference genome here is actually not a very good reference genome, because when we take our sequencing reads, they look quite different from the actual HLA alleles this patient harbors. So when we take our reads and try to align them, essentially they don't align to this region of the genome. So we get very poor coverage in this region of the genome, which means we can't really infer whether or not we've got LOH or not. And this is what's hampered a lot of previous studies, is that you can't infer the copy number of the HLA region. So we decided, well, we can take quite a simple technique, because we know the HLA alleles of that specific patient, because we can use the sequencing data to do this. Why don't we align the reads specifically to the HLA alleles of that patient, the maternal and paternal alleles? And so this is exactly what we did. We developed a method that does this. So we take the normal reads and the tumor reads and align these to this patient-specific HLA alleles, and this then allows us to try to work out the copy number of the HLA alleles. And we've developed a method which we call LOLA, for loss of heterozygosity in human leukocyte antigen. And this is an example of one of the patients where we looked at this. We can see the two different HLA-A alleles. We're looking at the difference in tumor to normal coverage. We see a clear difference here between the two alleles. And we can look specifically now at the heterozygous positions where these alleles differ and see whether we see differences in the log R in this case. And we can get a simple p-value to explore this and say that these alleles are clearly imbalanced. And then we can also integrate the purity and ploidy to try to work out the integer copy number of these alleles. And in this case, we clearly see we've got 2 copies of one of the alleles and 0 copies of the other. So we've got a tool which we think works to allow us to infer the copy number of the HLA allele. Now the question is, how frequently do we actually see these events? So we first of all looked in our TRACERx cohort, and in this case we found actually 40% of the patient's tumors appeared to have HLA-LOH at least one of the regions. So this appears to be a highly prevalent mechanism potentially of immune escape. And we see it's more frequent in the lung squamous cell carcinomas than it is in the lung adenocarcinomas. So then we can ask, when does it actually occur during tumor evolution? Is it an early event or a later event? Well, here we can map this to the tumor's phylogenetic tree to work out whether it's clonal or subclonal. And we found in some cases we could clearly map it as a clonal event occurring early during tumor evolution, whereas in other cases we see it as a subclonal event occurring later on. And interestingly, in certain cases, we actually think there's evidence that it's occurring in multiple times during tumor evolution, suggesting there's really quite strong evidence of positive selection, that we're seeing it occurring independently on separate branches of the tumor's phylogenetic tree. But then we want to ask, is it in any way associated with metastasis as well? And here we obtained a cohort where we had primary lung tumors and then paired brain metastasis. And we could ask, is it subclonal only in one of the primary or the brain, or do we see it clonal in both cases? And when we looked here in this cohort, we could see that as well here frequently it was a subclonal event occurring only in either the primary tumor or the brain metastasis. when we specifically looked at this, whether it was enriched in the brain metastasis compared to the primary tumor, we see a certain enrichment for it to be, when it's subclonal, it to be more frequently to us only identify in the brain metastasis and not in the primary tumor. So we think it's occurring frequently, and it's frequently a subclonal event, but ultimately want to ask what is the functional impact of this HLA-LOH event? Well, as I said, losing an HLA allele may diminish the capacity to present neoantigens. So these might no longer be presented, and so therefore the T cell no longer recognizes that cell as foreign. So one thing we could ask is, do we actually see differences between sister subclones where one clone has lost the HLA allele and one clone hasn't? We predict potentially we'll see more mutations in the clone with the LOH event because more of these mutations are essentially become invisible to the immune system. And so we looked at exactly this, and when we looked at the LOH clone compared to its paired no LOH clone, we saw a clear significant difference with more mutations in the LOH clone compared to its sister subclone. There was only 2 cases where we saw this went the other way. And we could also see whether there was enrichment for neoantigens binding to the lost allele compared to the kept allele. And when we did this, again, we saw a clear trend for more neoantigens binding to the lost allele, so these neoantigens potentially no longer being presented to the immune system. And when we actually predict this across the cohort, we can see that the number predicted to the lost neoantigen, we've actually got, we can have a few thousand of neoantigens that are no longer being presented, which really raises questions if we're trying to design some sort of TIL therapy or vaccine therapy where we want to predict neoantigens, we really need to take this into account because many of those neoantigens may no longer be presented. And now, okay. And then one question we could also ask was, so what happens, is this occurring in response to an active immune environment, or does this lead to immune depletion? So here we looked at the immune microenvironment in tumors with HLA-LOH. And the first thing we looked at was PD-L1 staining on the diagnostic block, so we could only look on the tumor as a whole. And when we looked at this, we saw tumors with a clonal LOH event. We saw the immune cells and the tumor cells stained for far higher PD-L1. So we think there's other mechanisms of immune evasion here. This is an active immune environment, which therefore is selecting for loss of the HLA allele. We see a clear difference when we don't see any LOH. We see less of an active immune microenvironment. So we next asked, okay, so we've seen this in the TRACER-X cohort. Can we also validate this in other cohorts? So here we looked in the TCGA as well, and here we could see in both lung adeno and lung squamous cell carcinoma, we can predict these HLA-LOH events, and again we see it enriched in the lung squamous cell carcinomas compared to the lung adenocarcinomas. And here again we see an enrichment of various measures of immune infiltration when we see this HLA-LOH event. So we think it's occurring in response to an active immune environment potentially. Okay, so that just leaves me to conclude. So I hope I've convinced you that, and as we've seen already, that sort of tumor heterogeneity is fairly widespread, particularly in these early stage non-small cell lung cancers. We see it occurring at both the mutational level and at the copy number level. And we think that this copy number heterogeneity appears to be particularly prognostic, although I think there's clearly more work to explore more detail to the extent to which the clonal architecture may be prognostic in these tumors. And I haven't actually talked about that, so genome doubling, I'll leave that for another time. And then we can also see that clonal neoantigens potentially hold prognostic relevance here, both in the treatment-naive setting and in the context of immune checkpoint blockade. And then we think this HLA-LOH potentially provides a mechanism of immune escape in these lung cancers. And we're now looking at other mechanisms as well, and we see that most of these tumors appear to have some sort of mechanism of immune escape. So we think that the immune system is really one of the key forces which is shaping the tumor landscape that we see even in these early stage tumors. So that just leaves me to thank everyone that's been involved with this work as well as you all, and I think there's some time for questions as well.
Thanks. So you were looking at genetic event, which is LOH in HLA, but you can also look at the RNA-seq data to see the expression changes. And maybe you can have even stronger effect if you just look at the expression, and you should have expression data for your tumors, right?
Yes, that's a good question. So we've started looking at this, but we haven't looked in. So initially we looked at the RNA-seq without mapping to the patient-specific HLA alleles, and so there we see there is some downregulation of HLA as well. But now we're moving on to try to do this where we're essentially integrating our tool to allow it to use RNA-seq as well. The slightly tricky thing is you can have downregulation, but that we can't tell whether that's because of a loss event or not. So we need to integrate that with our copy number tool as well. But I think that's a good point. Yes. Yeah, I think it'll be very interesting to see how many times do we see sort of what looks like down HLA allele where at the genomic level it seems perfectly intact.
So I'm wondering, are some of these effects predictive of immune response or survival? How much can you do with kind of proxy effects? And what I mean by that is, loss of heterozygosity of the HLA alleles is predictive of poor immune response. How much would you get just from knowing which tumors are prone to copy number aberrations versus not? And taking it a step further, copy number aberrations are predictive of pure response. How much would you get if you just stratified by the P53 mutated versus not tumors?
Yeah, I think that's a good point. I mean, I think the data here, one of the tricky aspects that in terms of exploring the relationship in general between copy number and immune response and immune infiltration is that when you've got more immune infiltration, that also necessarily means the tumor purity is lower, so the ability to actually detect the copy number events goes down. So I think in general, and I think that's been an issue with some of the studies that have looked at a sort of broad relationship between copy number and immune infiltration. And when we do see, so one of the things that's quite interesting is we don't see the association with immune infiltration and the mutation burden sort of in the HLA-LOH, we don't see it when we just look at allelic imbalance of the HLA, specifically when there's loss. So we think it's not just a question of having more copy number aberrations. And I think in some of these cases, it's sort of the HLA loss stands out as a very focal event, whereas in other cases, it probably is more reflective of just ongoing chromosomal instability.
So if I understood the correlation you showed for neoantigens, it seemed like there were a lot of neoantigens that were clonal, and this was potentially a good thing for immunotherapy. Is that right? Like that, and then so if so, that sort of raises the question of why these cells aren't killed by the immune system if they're presenting a lot of neoantigens. And this Hannah Carter paper from last year actually had the opposite correlation, where the claim was that there were actually few neoantigens that were presented because the immune system would have given. So like the idea would be most driver mutations are not actually presented by the patients containing them. So it's sort of the opposite correlation. I wonder if how that sort of affects your analysis, or how to square these two things.
So I guess the first, so yes, we definitely see that. Well, we think there's a lot of predicted neoantigens. So I guess that's the first thing, that a lot of these aren't true. So we're predicting too many, and probably quite a lot of false positives. The second part is, so why are we still seeing tumors? So you might predict if we have that many neoantigens, then we shouldn't be getting to the stage of having a tumor. And I guess I think that partly relates to the fact there are various different escape mechanisms. So one of the common ones is sort of upregulation of these checkpoint molecules. So we see, so in the context of immunotherapy, I think they wouldn't benefit unless they had the immune checkpoint blockade. So I think that, so there we're actually allowing the T cells to no longer be in the exhausted state. Yeah, so I think there's probably multiple mechanisms of immune escape occurring and I guess it depends on how early it needs to be, and how many cells, is there a critical mass before the immune system sort of kicks in. And I guess in terms of, I think what we're seeing here is still a very, I think sort of agrees with the Carter paper that these tumors are quite heavily sculpted, and there's a lot of immune editing going on by the time we see it. And this isn't, that these are, although we see a lot of clonal neoantigens, these are probably far fewer than there could have been if there hadn't been an immune system in place, would be my hypothesis. I'd suggest they're somewhat, so I mean, I guess ultimately what we're suggesting is that the immune system's definitely, because I think this has been a contentious issue, whether in early stage cancer is the immune system at play or not. And I think I'd suggest that both sets, sort of our analysis and their analysis, suggest that yes, the immune system is playing a role in sculpting the tumor at an early stage as well.
So you said that the mutation load of point mutations was not predictive of patient survival. Did you also look whether subclonal driver gene mutations were predictive of survival?
We didn't see a clear relationship there. I mean, I think there's, to be honest, I think we haven't sort of mined this in enough depth, and we sort of set, before the study started, we said, okay, we're going to explore these. We'll look at copy number heterogeneity like this and mutational heterogeneity, so it's a sort of prospective study, so we're not sort of influenced by, because we were a bit worried that we'd look at too many things and then find something very exciting and then report that, and so the moment we've sort of tried to stick, and we're now able to start looking a bit more in this 100 cohort, and then we can use the rest of the cohort as a validation set. So I think we're trying to start looking into these things in more detail, but at the moment it's quite preliminary, I guess.